Triton and SDS are three common detergents we use in the lab, which we use for more than just cleaning up a mess. We use detergents for all sorts of things, and there are lots of different detergents. So today I want to talk a little bit more about how to choose which one to use.
Detergents are a type of surfactant or surface acting agent, and they get this name because they're able to act at surfaces such as air-water interfaces or water lipid interfaces. So where you're going between something that's hydrophobic or water avoided and hydrophilic, water loved. And they're able to do this because they have hydrophilic parts and hydrophobic parts too. So they're able to disrupt membranes that are made up of phospholipids and kind of work their way in there. They're also able to interact with both the hydrophobic and the hydrophilic parts of proteins. So we can use detergents for a lot of different purposes. And some of the main things that we use them for in the lab are breaking open cells, so lysing them by disrupting those membranes, extracting membrane proteins in a water-soluble form, preventing proteins from aggregating by kind of like protecting those hydrophobic parts from clumping together, preventing non-specific binding, so just proteins just kind of binding to surfaces because those hydrophobic parts don't have anything else to hang out with. And so this would be done in like pull down. So when we're doing things like immunoprecipitations, I'll get more into some of this stuff in a minute. Various blots, Western blots, etc., when we want to prevent non-specific binding. We can also use harsher detergents for actually denaturing or unfolding proteins, such as when we're doing SDS page, which is a technique that lets us separate proteins by size or by their length because they're unfolded in order to kind of like see what size proteins are in a solution and how pure it is and things like this, or before a Western blot. And I have much more on all these things in other posts, but this is just an overview of some of the things that we can use detergents for. But which detergent to use for what? We use different detergents for different purposes. These are just a few of the ones that we commonly use in biochemistry, but there are many more. And if you go on Google, search for detergents, you'll do like biochemistry detergents or something like that, you'll find a lot of different ones. And you can see that this is another non-exhaustive list, but of some of the common ones that are used. We're going to go over how we can kind of like interpret a table like this and help choose which one to use. What do these different properties mean, things like this. Before you get too scared away by all of these different properties, know that in addition to, we're going to go over them in detail. This pretty much all you have to worry about is usually whether something is ionic, zwitterionic, or non-ionic. And really, you don't need to worry about it usually that much, because what you're going to do is you're going to go with a protocol and see what it uses. Because sometimes different proteins might have different peculiarities where they like different ones more than other things. And so your best bet is to start with the protocol. But then it's always good to know why you're using certain things. And then if things aren't working out, be able to experiment a little. But you probably don't want to go between different classes. This is because the ionic ones are going to be harsh, whereas the non-ionic ones are going to be much milder. And the zwitterionic ones are kind of somewhere in the middle. So by harsh, what I mean is they're going to be like denaturing. They're going to disrupt the intra-protein interactions to like unfold proteins and disrupt inter-protein interactions to break up protein interactions. Whereas the non-ionic ones, they're going to be milder. They're going to typically avoid breaking up those crucial protein interactions, and instead just break up the lipid-lipid interactions and the lipid-protein interactions, allowing you to say extract a protein from the lipid environment without harming it and without separating it from the other proteins that it's hanging out with. And you can also use these to like break open cells, maintain everything in the state that it's in mostly. So, bionic, I just basically mean charged, and non-ionic is not charged. And zwitterionic means something that has charged parts and non-charged parts, but they cancel each other out. And in all these cases, the charged part that we're talking about is or the non-charged part we're talking about, what we're going to be talking about is we're going to be talking about that hydrophilic head. So the hydrophilic head is going to be the part that's going to allow the detergents to hang out with water. And the hydrophobic tail is going to be that hydrocarbon part that's going to allow it to hang out with the lipidy stuff and with other tails. This head is always going to be hydrophilic, but it may or may not be charged. If it is charged, like an example of this would be SDS. This is anionic, so it's negatively charged. There are also ones that are cationic or positively charged. With something that's zwitterionic, you're going to have both positive and negative parts in the same molecule. I'm in that same head group, so you can see you have this negative charge here and this positive charge here, and so those cancel one another out. And then when we talk about non-ionic, things like tween and triton, these are not going to have any charges, but they're still going to be hydrophilic in those head groups. Also note that kind of like the bulkier the head group and the longer the tail, the more mild it probably is. It's going to be harder for it to just go into proteins and disrupt them and things like this. And when you have something that's ionic, it's going to be able to disrupt more things than if you have something that's non-ionic. So if we want to do something like denature a protein, so like unfold the protein, such as for SDS page, here it's really important to use one of those harsh detergents that's actually going to break up those protein interactions and unfold the protein. And because it has those hydrophobic parts, it can kind of glob onto the hydrophobic parts of the protein that you've exposed and keep it soluble. And the charge is also going to provide the charge you need to run through the gel. So anionic detergent like SDS is great for that. But if you're doing something where you actually want to, say, just break open the protein, break open the cells and protect the proteins, or you want to do something like a co-immunoprecipitation or other sort of pull down where you want to see what proteins are interacting with other proteins, but in a specific way, you don't want them to just be binding anything. So you want to reduce that non-specific binding. Or if you want to really preserve a protein in as native as possible state it can be, with these things, you're going to want to go with something that's going to be very mild. Thermo has this kind of like table of some that are commonly used. So we can see that we have the SDS we talked about, which is going to be that strong one, that denaturing one, that harsh one. Then we have other ones like triton and tween that we might use for lysing cells and doing things where we care about the proteins. And they have a chart if you want to learn more about which ones you might use for what different purposes. Within a group, though, you can see that there's a lot of different ones that can be used. Some of it's just going to depend on what that protein likes. And so you might need to actually take some trial and error to figure that out. There's also some specific things that you might want to keep in mind, such as that some of these detergents are actually going to absorb UV light. And so an example of this is triton. So triton has this aromatic group that's going to absorb UV light. And often we use UV light absorption, especially at 280, in order to detect proteins and even figure out protein concentrations. And so if we have detergent in our solution that contains the protein, this is going to mess up that analysis, especially if you don't blanket well. So you might want to use something like tween instead of something like triton if you're trying to analyze where a protein is, say, coming off a column in chromatography. So now let's go into a little more detail about how you can choose based on other properties. And before you get too scared away by this slide, we're going to go into them one by one, but I just wanted to give you an overview of some of the things that we're going to be looking at. And each of these properties is going to vary for different detergents and under different conditions. So things like temperature, pH, all of that different stuff is going to affect these. And then you have to choose which one's going to be best for your experiment, which again might take some trial and error to find out. And your best bet is to start with a published protocol if available. So to understand what these different things are, let's step back and understand what's going on when we have phospholipids and detergents mix. So both of these are amphiphilic or amphipathic. They have both hydrophilic and hydrophobic parts. And so as we talked about, they have those heads that are hydrophilic and may or may not be charged in the case of a detergent. They are charged in the case of a phospholipid. And then they have those lipid tails, which are hydrophobic. Now, phospholipids, they have two tails and detergents have a single tail. And this has a big consequence because what's going to happen is these detergents are going to be kind of cone-shaped, whereas the phospholipids are going to be more of a rectangle. when you stick one of these amphiphilic molecules into a watery environment, what they're going to do is they're going to organize themselves so that their hydrophobic parts, those lipidy tails, are going to be together away from the water, and their hydrophilic parts, the water-loving parts, are going to be exposed to the water. For a phospholipid, because of that rectangular shape, the best way that it can do this is to form a bilayer, where you have a sort of sandwich where the head groups are going to be like your bread, and those tail groups are going to be like your peanut butter. But for a detergent, it has that cone-like shape, so what it can do is it can form these single-layered micelles, and it can form these kind of like bubble-like things where those lipidy tails are in the center and the hydrophilic heads on the outside. And if you were doing something like washing away gunk, that gunk would be trapped in the center as well. But hopefully we don't have too much gunk in our protein mixtures. What's going to happen when you add a detergent to a cell which has a phospholipid membrane? That detergent, because it's amphiphilic like that phospholipid, because it has a shape kind of like that phospholipid, it's going to kind of worm itself into that membrane. And this is going to disrupt the membrane and break open or lyse the cells. At a higher concentration, it's actually going to be able to kind of extract the membrane proteins from the membrane and pull them into their micelles, allowing these proteins to stay soluble in watery environments. So in a membrane protein, that part of the protein that actually spans the membrane is going to be hydrophobic because it's hanging out in that lipidy environment. And so you need to protect those hydrophobic parts when you extract them, and the micelle is able to allow it to do this. But you need enough of the detergent molecules in order to form these micelles. And there's a value called the critical micelle concentration, or CMC, which is the concentration of a specific detergent at which those individual detergent molecules start teaming up to form these micelles. And so the CMC is going to be one of the main properties of different detergents that you're going to be looking to when you're trying to figure out which one to use. So the lipidy situation is going to depend on your detergent concentration. So the first step is just going to be disrupting those membranes for lysis. So at a ratio of like 0.1 to 1 to 1 to 1 of detergent to your membrane lipid, the lipid bilayer is going to stay mostly intact, but you might be extracting some membrane proteins. And so some membrane proteins are going to be extracted more easily. And so you might start extracting some of these proteins at the lower detergent ratio. As you raise the detergent concentration, the membrane gets solubilized and you have mixed micelles, which are going to contain various proportions of detergents, membrane lipids, and membrane proteins. So you're going to have a mixture of all sorts of different micelles, which are going to have a mixture of all sorts of different molecules. But if you go all the way up where you have like a 10 to 1 ratio of detergent to lipids, you're pretty much going to have replaced all of those membrane lipids with detergent molecules in your micelles. And now you have these nice solubilized membrane proteins. Unless you're trying to actually extract proteins into those micelles, you want to stay below the CMC so that you're not going to have those micelles form and say partition off some of your proteins in your mixture. So typically you're going to be working at really, really low concentrations of detergent. So for example, if you were using something like tween 20, you're going to want to stay below 0.06% weight volume. With a Triton X100, you're going to want to be below 0.24% weight volume. But if you're trying to use detergents in order to like solubilize a membrane protein, well, here you're going to need to be above your CMC. So you're going to have higher concentrations that you need. And as we'll see, the CMC is also going to come into play when we think about how we remove excess detergent. So we talked about how CMC was one of the main properties. Another one to look at is aggregation number. This is going to be the average number of detergent molecules in each of those micelles, which kind of goes hand in hand with the micelle molecular weight, which is a measure of micelle size, which is going to depend both on that aggregation number as well as the size of the individual detergent molecules. So these molecules can vary in size. The cloud point, this is the critical micelle temperature, or CMT. This is the temperature at which at and above which the detergent micelles are going to aggregate, separating into two phases. So you're kind of going to get this cloudy phase, which is detergent rich, and this clear phase, which is detergent depleted. And this is going to be a way where you can kind of bulk separate the detergency stuff from the non-detergency stuff in a solution. And so if you're working with a protein, say you'd want to have something with, and you're trying to do this phase separation, you'd want something with a low CMT so that you don't denature your protein when you have to go to a higher temperature in order to phase separate. If you want to know more about it, here's a nice overview type thing. So you can see that as you raise the concentration, you're going to get more micelles. And as you raise the temperature, you get to this point where you get this phase separation. And the individual things are going to vary based on different types of detergents and things like this. And it can get really complicated. But just know that at or above the CMC, you're going to get these micelles form, and at or above the CMT, you're going to get phase separation for like non-ionic detergents. And so you can see all the different properties of these 4 different detergents that you might be using in the lab. And so, for example, you can see that for something like this octothioglucoside, it has a pretty high CMC, as well as like CHAPS. So this means you have to get it at really high concentrations in order for it to form those micelles. But if you have something like tween, this is a really low CMC, so it's going to form those micelles really, really rapidly. As a result, well, it's also going to make it harder to remove the detergent. So detergent can be great for some things, but it can also interfere with other things. And so you don't want to use a super high concentration of detergent in a lot of different experiments. And so you might want to at least lower the concentration of detergent, if not just like remove it. And there are different methods you can use to do this. But if you have a low CMC, it's going to be difficult to remove it because those micelles, you'd have to get the detergent to a really, really low concentration in order to break up those micelles to be able to get rid of that detergent. And so for something like that, you're not going to want to use a method that relies on those micelles breaking up and dialyzing out, or using size exclusion or something like this, a size-based method where you're going to need those individual monomers to be separated in that form rather than actually kind of trying to remove the micelles themselves. For a method where you're kind of relying on the proteins binding to something, such as with a chromatography method. If you're using a chromatography method like ion exchange chromatography, well, this is going to work by binding proteins based on charge. And you don't want to use this if you're using an ionic detergent because, well, the ionic detergent is going to bind to that ion exchange resin as well. And this is going to make it so that you can't remove the detergent in this way. As I mentioned, a common way to remove it is dialysis or size exclusion chromatography, which is going to basically use these resins, these little beads that have these pores. And if you have something that's really small, the detergent molecule will get stuck in the pores. It won't get stuck, but it'll travel through all these little pores in the beads, and it'll have to go slower than the bigger things, which can't get into those tunnels in the beads, and so goes around and comes out sooner. So bottom line, you're separating things based on their size. If you have really big micelles, and those micelles aren't coming apart, like dissociating into individual detergent molecules because you have a low CMC, or even at low concentrations of forming these micelles, it's going to be hard to remove it by one of these methods like the gel filtration, like the size exclusion, or like dialysis, where basically what you have is you have a solution in a little pouch, and big things can't get through the pouch, whereas little things can. And so you're going to then kind of stick that little pouch into a big beaker or something of liquid, and the small things are going to diffuse out, they're going to wander out, whereas the bigger things are going to be trapped. And in order to remove detergent in this way, the detergent has to be small enough to get out. And if you have those really big micelles and they're not coming apart, those are going to be hard to get out. So if you look at, say, a chart like this, you can see that a lot of the ones we commonly use, so Triton and Tween, etc., these are going to not be dialyzable. There are methods that can be used to remove certain detergents, and it's like proprietary things and like that. But yeah, just something to keep a note of. So the exact detergent you're going to choose is going to depend on things like the various properties. But again, you're usually starting with some sort of protocol. So people have figured out what works best for various types of experiments. And so you often want to just at least start by going with that. It's really important to know, though, the differences between like a harsh detergent and a mild detergent. So harsher detergents are going to be like ionic detergents, things like SDS. They're going to break up the protein-protein interactions, the intra-protein interactions, so denature or unfold your proteins. Whereas something non-ionic, like Tween or Triton, this is going to be milder. This is going to only break up like lipid-lipid and lipid-protein interactions. And so you're going to want to use something like this when you're doing something where you're trying to preserve protein-protein interactions, or you're trying to preserve the protein in its most like natural state.